Okay, everybody. Assalamualaikum. A very good morning to all members of Persatuan Pegawai Sains Malaysia and participants from overseas, I hope. So, I'm Jane, again, your moderator for this lovely morning. So, today, our Scientific Webinar Series 2025, which is organized by Persatuan Pegawai Sains Malaysia, will bring you another exciting topic to be discussed, entitled Leptospirosis Insights and Further Prospects. As we all know, leptospirosis is a significant public health issue in Malaysia, with transmission through animal urine, occupational exposure, and recreational activities being a major contributors. Okay, so early diagnosis and treatment are crucial, and prevention strategies, including hygiene practices, rat control, and avoiding contaminated water, are essential to mitigating the risk. So we are very lucky today to have with us our speaker from Department of Medical Microbiology Faculty of Medicine and Health Sciences, Universiti Putra, Malaysia. That is Professor Dr. Vasantha Kumari Neela. So our scholar graduated with Bachelor of Science in General Microbiology from University of Madras and obtained both Masters of Science and PhD in Medical Microbiology from Universiti Putra, Malaysia. So she is currently an Executive Council member of International Leptospirosis Society since 2024, and nevertheless involved in international recognition professionals, qualifications, membership, affiliate, which are mostly in the areas of interest regarding leptospirosis. So, ladies and gentlemen, without further ado, we are very proud to welcome Professor Dr. Vasantha Kumari Neela for her presentation. The floor is yours.
Thank you, Ms. Jean, for the kind introduction. And also thank you for the opportunity to share what we have done on leptospirosis with a big audience here. So I guess the audience here are mostly from the science officers and from the MOH. And so I will try the best how I can enlighten the knowledge on leptospirosis. Okay, so as Ms. Jane said, my name is Neela. I'm in the Department of Medical Microbiology, Faculty of Medicine and Health Sciences, UPM. So we started working with leptospirosis since 2015, and it is like a decade now. Before that, I was working mostly on the drug-resistant pathogens. Even now, I do work on that. But for the past 10 years, we are concentrating more on leptospirosis because it is a disease of concern in Malaysia. Okay, so let me go through the basics of leptospirosis. So this talk will cover what is leptospirosis or what is leptospira, and how can we control, or how can we reduce the incidences. This is a very a preventable infection without a vaccine, so it can be prevented. So I'm going to brief later how can we reduce the incidence rates. Okay. So many of us know what is leptospirosis, but since we are in the field, but when we ask outside, especially when I ask my undergraduate students, new students, or outside population, they will not know leptospirosis. But when I say rat's urine disease, then they will know. So it is very well recognized as rat's urine disease among the public. So now we need to know how this leptospirosis, or how the human is getting this leptospirosis. Firstly, it is a zoonotic disease. So when we say the word zoonotic, we know that it is a from animal to human. So the word zoonotic says it is from animal to human. And this leptospira is a spirochete and it is a bacterial infection. It can affect both, though it's a zoonotic, it can affect both human and the animal. Though it can be found in many animals or mammals like cattle, pigs, horses. For many animals, they have studied and found a leptospira antibody or leptospira in the animals. But the most common reservoirs, or the one that always actively participate in the infection, is the rodents. So rodents are considered as a main agent for causing leptospirosis. So animals serve as the vectors, shedding the leptospira in their urine for several months without any signs or symptoms. So the difference between human and animal is animals, especially rodents. In rodents, these leptospira is more like a normal flora. I mean, not a normal flora, but they can carry without causing any infection. So in rodents, in the kidney, they can keep on multiplying, and that will be excreted through urine to the environment. So for the rodents, it does not cause any infection. It is more like they are just carriers. But for other animals like cow or livestock, they do cause some infections which can result in abortion and others. But when we look at Malaysia scenario, I don't see livestock is a major problem. It is not, lepto is not that much affecting the livestock. But if you look at countries where livestock is a major economy, like New Zealand, Australia, all those countries where the livestock population is more than the human population, they have to really keep a check on the leptospira, because it is the economy. But here, talking to my vet colleagues, they said that livestock, it is not a big issue, although we see some antibodies, but it is, so far we did not see any very big issue with livestock. Okay. So they just multiply in the kidney, and then it will be excreted to the environment. So how it works, how the transmission happens is, from the animal, it is transmitted to the environment through the urine, and human, when we get exposed to that contaminated soil or water, we have the chances of getting the infection. So if you look at this picture, you can see the cow, you can see the rats. Okay, all these are this is just a created figure by our group. So here you can see that a human swimming or in the water. This is not a swimming pool. We just assume that it is like a recreational, like something like a recreational forest or recreational water area. So here we can see that the animals urinating. They come here to get food, and they urinate, and that urine will contaminate the water here. So human, when they are enjoying their recreational activities in this water, they tend to drink or swallow. I mean, they tend to swallow water, or if they have any cuts or anything on their skin, so this organism can enter their body. That is how the human get exposed. Okay? So I think you have some idea how human get this infection. Okay. So it is usually through the cuts, inhalation, or ingestion. Okay, so this is again a little bit more detailed explanation of the previous slide. So you can see here, Soil, water, and vegetation. So this is like commonly shared by human or animal or whatever it is the environment. So here the rodents come here because sometimes, like most of the time, the rodents come here in search of food. Because in the recreational area, when people go, they just throw the food or so these rodents come to eat the food, and while eating, they will urinate, and that urine will contaminate the soil or the water. So the water is like flowing. So the water can, the leptospira can be like through the water, can go from one area to another area, and it can settle somewhere. So when human come in contact with those area, they can get the infection. Okay. So what it can cause in human is, maybe I will go later, but in detail, but you can see some picture, some infection types. Like it can cause infection is like a multiple organ infection. So it can cause meningitis, UITs, pulmonary hemorrhage. This is the most common one, pulmonary hemorrhage, myocarditis, hepatitis, nephritis. Okay, so these are some of the identified infections that leptospirosis can cause in a human. And in animal, also it can cause infections like abortion, as I said earlier, hepatitis, nephritis, stillbirth. So these are some of the reports from other countries. So they can they can affect the livestock or dogs. But we do have like infection of dogs in Malaysia. But I don't see much in the livestock. Okay. So animals like rodents, cattle, all these are the natural host. So that when we say natural host, that means they can live there happily without being affected by the immune system of the host. So they can happily live, multiply, and then get excreted through the urine. So they are not affected by the any external force. Or they can also live in the environment like water, soil, everything. But human becomes the accidental host. So when human becomes the accidental host, they don't come purposely to human, but it happens that they come to the human, and then where we are the accidental host, but when it comes to the human, our body will start fighting with them. When we fail, they will cause infection in us. Okay. So going to the organism, so it is the organism which causes leptospirosis is Leptospira. So we have many types of Leptospira, which are like, we can classify them in saprophyte, intermediate, and pathogenic. So the saprophytes are the intermediate, are not a really worrying thing, but the pathogenic Leptospira, they are the one which is actually causing the infection. Though we have like many species, like more than 64 species, it is being updated. But only like few species that are pathogenic are really harmful to the human. Others, they just live in the environment and they are not very harmful. This is spirochete. So you can see the shape, it is like a spiral shape. And it is very thin. The organism is very thin. The diameter is only about 0.1 micrometer, while the length is 10 to 20 micrometer. So it is very different from the other bacteria because it is a spiral shape, and it is very thin, and which helps them to move very fast, and it has like a hooked end. They have a hooked end. So when you have a hooked end, it can hook to one the back-to-back Leptospira. So you can sometimes see a very, very long spirochete, long bacteria, but it is actually like a hooking 3 or 4 or 5 Leptospira together. Okay. Okay, so it is a saprophyte that lives in the environment. The growth temperature it is usually like 28 to 30 degrees. So if you look at the normal other bacteria that we grow in the lab, it is usually 37. But this particular organism, we need to give a specific 28 to 30 degrees for them to grow. So we always have a dedicated incubator in the lab just for Leptospira, because we cannot grow this in the other the common incubator, because that is always set at 37. Pathogenic strains live in the kidney of host animals. So here we need to underscore this, because though we have saprophytes and intermediate, but most of the time the pathogenic strains come from the animal, like the rodents or animals. So that whatever we get from the environment are usually not the pathogenic strains, because the one that grow in the environment, they are like most used with the environmental conditions, and they can grow even at a lower temperature, like 10, 15, less than 20 degrees. But these pathogenic strains, it always comes from the animal, like mostly from the rodents. And what makes them pathogenic is the outer membrane. So this is a gram-negative bacteria, and it has an outer membrane, and this outer membrane has we know that outer membrane is always made up of lipopolysaccharide, and it has several other outer membrane proteins, which are important for them to adhere to the host tissues, and also um, make our body to produce immune response. Okay? So if you look at this picture here, we try to do some silver stain to see, but the most important with this organism is we cannot do gram stain. It doesn't take the gram stain. So staining is very difficult. Staining is difficult in this organism. So we only look at, use a dark field microscopy, where we take little bit of the culture and put in a slide and view under the dark field microscope. So you can see the spirochetes in this form. So gram stain will not work. So we have to do this in order to identify Leptospira, you definitely need to have a dark field microscope. Okay. Okay. So a quick understanding on the worldwide morbidity and mortality. This is a study that was done, if not mistaken, on 2016 by Costa from Brazil. After that, there is no study on there is no report on what is the worldwide morbidity and mortality. So we have to still use this data. Maybe it is higher or lower, but we are yet to get the new paper on or new data on this. So if you look at this scale, we can see that this India, Malaysia, these regions, Malaysia comes here, these regions, we have a lot of cases. And this South America, especially this region, have many reported cases. So it is very common in the tropical area, but it does not, those which are like yellow or, it does not mean that we don't have cases there, but maybe there are not enough studies. Maybe it is not a big issue there, like they don't see like infection or death from that, so they did not look into that, but it is very like endemic in many countries. So if you look at the data, about 1.7 million per year with 95,000 deaths. So, and 65,000 death comes from the Asian region alone. So although it is like distributed worldwide, but when we look at the death cases or the mortality cases, it is more common in the Asia. But when we just compare with, I just had this extra point to say that dengue is very common and more than 100 million infections per year. But the death rate is only about 200,000. But when you compare with lepto, it is 1.75 million, but the death is higher. So that's just for a comparison, just to tell how serious the illness. Okay. Clinical manifestation. So now, leptospirosis can be classified into two types, anicteric and icteric. When we say anicteric, it is self-limited, headache, cough, non-specific flu-like illness, muscle pain, fever, diarrhea, rarely fatal. But icteric, which is actually the severe leptospirosis, it can cause this Wales disease, where you can see renal failure, jaundice, hemorrhagic fever, and respiratory distress, multiple organ diseases, like it can affect the heart, CNS, muscles, etc. And it can be fatal, and recovery takes a lot of time, like it takes months to recover. So, since there are like both scenario, like icteric and anicteric, only about 10% of the cases are severe. But 90% of the cases are not severe, but it will be like we will not even know that it is leptospira, because the symptoms are quite common, or they share similar symptoms with other illness. So we may be exposed to leptospira, but since it did not really make us very sick, we will not go and test. Maybe we are exposed, but not all. Those who are having the risk factors, for example, like wherever we can get this, like who go for outdoor activities, or who are working in the farm, or who has like close contact with the rodents. So they may be exposed, but not necessarily that everyone get the infection. But at least about 10% can cause severe leptospirosis. So when we say severe leptospirosis, it is actually the Wales syndrome, which comes with the jaundice, hemorrhage, and acute renal failure. And then the next one is severe pulmonary hemorrhage, which causes acute respiratory distress. So what we see during the recent years, what we see commonly is that pulmonary hemorrhage, and the case fatality rates are more than 50%. The factors actually that determines the disease severity is the age, health, immune status, just like any other infectious disease. Age, health, and immune status contributes to the disease severity. Previous exposures, inoculum size, dose, the infecting leptospira. So the most important, apart from those common things, the infecting leptospira. So we need to know actually which, whether it is a pathogenic strain. If it's a pathogenic strain, which serovar. So this plays a very important role. So if you are just exposed to, because when we walk on with the barefoot on the soil or anything, we can get exposed because there are leptospira in the environment. But that may not be a pathogenic strain. That's why we don't get infected. So the infecting leptospira is very important to know whether you develop disease or not. A main challenge with this organism or leptospira is it is a biphasic illness. When we say biphasic illness, that means it has two phases, anicteric phase and icteric phase. In the anicteric phase, it is a first stage where like first 3 to 7 days, it will be like septic. That means the bacteria is inside your body. And you can get symptoms like very common symptoms like myalgia, headache, all those is common symptoms. But in the second stage, which is like 0 to 1 month, why we say 0 to 1 month, because you will actually start developing the immune response after 7 or 8 or 9 days later. But it says 0 because you may have like some antibodies from the previous exposure. And in this second stage, patients can develop meningitis, uveitis, and it can cause very severe if it is not like diagnosed and treated, it can cause severe infection. So what we see in the severe infection is the second stage, where like 10 to 30 days, the immune phase. So immune phase is like you will see all these antibodies developing, and that can cause like in this phase we can see the jaundice, hemorrhage, renal failure, myocarditis. So this is more like a dangerous infection, and this we can see in about 10% of the infected people. Okay, so this one is just to show that once the Leptospira enters the body through the skin or mucous membrane, it will go to the bloodstream, and from the bloodstream, it will go to the organs. So it can cause pulmonary hemorrhage. So this picture is actually our own study. So we did a study on the hamsters to see like when we infect the hamsters with the Leptospira, the pathogenic Leptospira, what happens. So we can see that hemorrhage, this dot, dot, dot thing is a pulmonary hemorrhage. Then we can see that acute kidney injury. Then we can see that hepatitis, and the brain, this is not our picture because we did not harvest, but we have studies showing that it can also go into the brain. Then other parts which we did not study, but from the literature we can see that can affect the heart, spleen, skeletal muscle. Clinical presentation. It can be multiple or non-specific clinical presentation. Non-specific or unique symptoms for leptospirosis. Typical Wales presentation is seen only in 10%, and prior antibiotic therapy will make them like not showing the symptoms. So the challenge with the clinical diagnosis is that there is no like a specific symptoms for lepto. They share similar symptoms with many other illness like malaria, typhus. You can see the renal failure in other infections also, meningitis, you can see in bacterial and other virus. So hemorrhagic fever, you can see in dengue, hantavirus, typhus. So it is very difficult to immediately make a decision or okay, this could be leptospirosis. That is a very difficult challenge because we are in a country where we have all these infections. So it is a bit difficult thing to, go for the diagnosis immediately, like a conclusive diagnosis. But patient history and epidemiological context plays important roles to be considered. When we say patient history, so we have certain history, certain criteria to suspect whether it is leptospira or not. So if the doctors will ask whether they had any recreational activity, or they were from any like do they have any contact with the rodents, or do they live in a very crowded environment where rodents come, do they have like exposure to rodents or animals, do they work in animal farms, so those kind of questions will be asked to the patients just to see whether there is any possibility for them to get leptospirosis. So that will help them. Like mostly like what we see in Malaysia is like post flood activities or post recreational activities. Okay. So laboratory diagnosis. So once in the hospital they have suspected or they want to go for some differential diagnosis, then they will go the laboratory diagnosis. Now we come to the laboratory diagnosis. So for leptospirosis, we can do direct examination, culture, serology, and molecular. But what will help in terms of treatment is very important. Direct examination, literature says that detection limit is 10 power 4 per ml of the blood. Bacterial load ranges from 10 power 2 to 10 power 6. The Brownian movement of the blood components and artifacts will affect the visibility and other spirochetes. So this direct examination, literature says that we can detect directly, but there are many, many challenges. The challenge is leptospira is like, it is very difficult that if you take one drop of the blood and put on the slide and see, you will not see much of leptospira because they are very like fast moving organism. They will be moving from here and there, and it is very difficult to catch them. And you must have at least like more than like 10 power 6 bacterial load so that you can detect, which is quite rare in that small amount of the blood. And even though you get like 1 ml, you're going to just put maybe 50 microlit or the maximum 100 microlit in the slide to see. So this technique does not really work. Also, the blood components and the artifacts from the sample will disturb the or your identification or your diagnosis. And sometimes, like other spirochetes, like if you are infected with other spirochetes, you are not sure whether it is leptospira infection or other spirochete infection. So this direct examination does not work for diagnosis, for treatment. Now, coming to the culture, many organisms in the hospital, we can just culture overnight and the next day you will see the colony like Staph aureus, E. coli, all you can see the colony on the next day. But with Leptospirosis, it's a very, very slow growing organism. Though they can move very fast, but they are very slow growing organism. They take maybe like 4 weeks to 13 weeks to grow. Or maybe at least minimum of, if it is like very fast growing, it will take at least minimum of 1 to 2 weeks to grow. And it does not grow in a solid medium. We need a specific medium. It's a liquid medium because it's a highly motile organism and they don't like this solid medium. So we have to, the medium is very specific for lepto. So it grows only in the liquid medium. And incubation is also like 28 to 30, as I said earlier. And so you need to have an incubator. So again, this will not work for clinical, like for patient treatment, because it is going to take at least like 2 to 3 weeks to 13 weeks. So we usually incubate up to 13 weeks. And after 13 weeks, if there is no growth, then only we discard the sample. So culture is not useful for diagnostics, but it is useful for like epidemiologic purpose, or if you want to isolate the organism, or you want to study something on the organism, or you want to identify what species, what serovar, then culture is useful, but not for treatment. Okay. So this picture or this video is how we can see the Leptospira in the dark field microscope. So basically you can see this one, like they are like together, like two Leptospira has joined together. So this is how you can see them moving in the dark field microscope. So to see this, you need the skill that person who are going to do this lepto, they should be really trained, because there are a lot of artifacts that you will see under the slide. So, and again, even in this one, to get this stage, like we have like 1 plus 2 plus 3 plus 4 plus. So we need to get this good sufficient growth. Only then we can see this 4 plus the number 4 plus means like we get more Leptospira that can be detected. So this is very important. So what we do in the lab is every week we take 1 drop of the medium and put on the slide, and then just look under the microscope. And then we see like, okay, whether there is increase in the number. That's how so once it has reached that 4 plus, then we make a stock. So how do we make the stock? We just, it's the same media, but we add a little bit of agar to make it as a semi-solid media, so that it is not very solid, but it is a little bit of semi-solid. And then they will just be stored in that. This organism, another challenge with this culture is we need to store at the room temperature, because they don't like very freezing temperature. They are very fragile, and usually like when we freeze for a long time, they will die, and it's very difficult to recover. But we did some studies to see preserve them in the minus 80. We can get them after we culture, we can get them live, but the recovery is very, very less. So what we do in the lab is like for some like safety, we do culture and store them in minus 80. But we do a regular, I think there are some questions, but I will keep the questions to the end so that we can discuss. What we do is every like 6 months, we subculture into new media. So we take a few, maybe about 1 ml from here. Then we subculture into new media. But before subculturing, we check whether there is no contamination. We check in the dark field microscope to check that there is no contamination. And then we subculture into the new media. That's how we maintain the culture in the lab. So every 6 months, we need to subculture them. Okay. So people working with Leptospira, it is a challenging organism that you cannot just culture and put on the freezer and just forget about it. But you need to subculture them every 6 months to make sure that they are maintained. And also the medium is specific and it is also a bit expensive. And okay, what we have, the traditional media contain about 8 to 10% of rabbit serum, Stewart, Korthoff, Fletcher, and Wood. So these are the like we call them Fletcher media because they also like made the media. So we have then they after these are the people like who initially started their own formulation of the media. But now what we commonly use is EMJH, which contain the bovine serum albumin, which is useful for the growth of most fastidious leptospires. It can be liquid or semi-solid or solid, but we don't use much of solid for storage. And it is incubated at 28 to 30 degrees. Growth is under visualization under dark field microscope. And 3 to 6 months we subculture. Again, this is not useful in rapid diagnosis, but important to get the leptospire isolates because the isolates are important for research. Okay. Okay. So now we know that direct examination will not work for diagnostics. Culturing also will not work for diagnostics because we cannot keep the patients waiting for 4 weeks to get the results to start the treatment. So what is commonly practiced in the hospital? We do serology. So there are serology, we have 3 methods that are commonly used for leptospira diagnostics, which is rapid test. This is very commonly used in the hospital. So basically, we have this IgM. We know that IgM is a recent infection. So if you have a IgM positive, then we can say that it is an infection. But this test is not very reliable because we also like sometimes like from the past infections, because we may be exposed to leptospira that we will not know. So we may have like IgM and the past IgG. So this, even though it gives a positive result, it is not conclusive. It is just for like a screening test. So once you have positive, then we can send to, we can do a ELISA, or even if it is ELISA positive, then we do a MAT. So MAT is called microscopic agglutination test, and this is actually the reference standard. We will not say it is a gold standard, but we can say as a reference standard. So those who are working with in the laboratory for leptospira, they will know what is MAT. So MAT is actually, not sure. Yeah, I think I have a slide on MAT. So serological detection by rapid test, we can use a serum or the whole blood. It can detect both the IgM and IgG with genus-specific antigen. But the challenge here is it is not able to identify the causative serovar or serogroup. But it is not very important to identify the causative serovar serogroup for the treatment. See, as long as it is infection, that is enough for the treatment, because this information on the serovar or serogroup is mostly for the epidemiological purposes. It usually takes about 10 to 15 minutes to get the results. So this is how you can see the results. You can do for IgG and IgM together, or what, because some companies which comes with only IgM, some companies with IgM and IgG. Okay. So when we get results for IgM, then we assume that it is a recent infection. But if we get only results for IgG, then we consider that as a past infection. ELISA is again, we can detect both IgG and IgM. But this is like a quantitative. The first one is only a qualitative test, but in ELISA we can do a quantitative test. Reaction of the antibodies with a broadly reactive genus-specific antigen, but again it does not identify the serovar or serogroup. And there are commercially available kits or in-house kits. And you just need a ELISA, a plate reader apart from the kits. Okay, so what is most commonly used across the world is MAT. Why MAT is used? Because this MAT actually tells us what serovar, what is the infecting serovar, and what is a titer. Because for leptospirosis, we have this reading, a positive result, a single titer of more than 400 is considered as positive, or fourfold or a greater rise in the titer between paired serum. That means serum on the first sample and serum on the second or consecutive samples. So what is this titer? If you look at this picture, this is a negative control. Then this is like titer. Your titer means you have different, maybe this is sample, not sample, the same sample, but different serovars. So you at one time you can test for many serovars, because you want to know which is the which serovar it is giving positive. Usually we test a panel of more than 20. We have a panel of more than 20 serovars. So what we do is we do a screening, like first initially we do a screening with all the serovars, and that means take the blood sample from the patient, take the serum out, and then the serum and the culture is mixed. And then if there is any agglutination, then you had to observe under the microscope. So this is a one-day test. So that you take the serum of the patient and you mix with the different serovar of the leptospira, and then you after 2 hours of incubation, then you observe under the microscope if there is agglutination for any of that serovar. That particular serovar, now we have to take and do the titration. So the stage 1 is actually, we want to check which serovar is actually interacting with the or agglutinating with the patient serum. So let's say we do a panel of 20 or 25 serovar, then and if it gives answers, the agglutination is given for like 2 or 3 or 4 or 5 serovars, then you just take that 4 or 5 serovars. Now the idea is to see whether it is a recent infection or a past infection, because this test actually, MAT actually identifies both IgG and IgM, because it is a whole serum. So that serum will contain both IgG and IgM. So now, so now what happens is, after you take those which has agglutinated and do a titration, the titration is done to see what is actually the at which titer you can see the agglutination. So most of the countries, like they have set 400 as the titer, but so if anything you see 400 and above, it is considered an infection, and anything you see below 400, it is not infection. And anything like you see like the day 1 when you suspect the patient is having lepto, you take a blood sample and you do a MAT. Then if you see like something like 50, 100 or 200, then you repeat the test after a few days to see whether there is any increase in the titer. So after a few days, if after 2 or 3 days, if you see that there is an increase like from 100 to 400 or above, then or like from 100 to 200, then you repeat again after 2-3 days and see whether it has gone up. That will tell you that it is a recent infection. This is how we do the diagnostic. So again, MIT is not a very good diagnostic test. It is again for epidemiological purposes. So the results are observed under dark field microscope, and we are able to identify the infecting or presumptive serogroups. So the biggest advantage of MIT is we can actually identify the serogroups that is causing the infection. And how do you get the serogroup? So most of the country which has uh, leptospirosis is like a common or notifiable disease there, they would have done some seroprevalence study. So they will always take samples from the patients or from the public to see like at what titer it is becoming infectious. Like for some countries, like where the cases are very, very less, even 100 or 200 is considered as infection. But some countries where it is very common, 400 may be like not considered infection, that maybe like 800 or above is considered infection. 400 is still considered as like, it is routine, it is common that people can have 400 even when they are exposed. So that decision or to make what is a titer for infection, it comes from the seroprevalence data. The seroprevalence is from the public, healthy individuals, risk people, and from the patient. So based on all these things, we can decide the titer that can be used for diagnostic purpose. Okay, so then comes the molecular. So we need to understand something. MAT or any serology will only work after 7 or 8 days of infection. That means after it has started producing the antibodies. In Leptospira, the antibodies are produced little later, like after 5 or 6 or 7 days only the antibodies start to produce. That is why like if we want to catch them earlier, we need to either get the organism, but culturing the organism is not a choice for diagnostics, but there is a good choice which is using the molecular technique or PCR. So if we want to catch earlier, then we can do a PCR from the blood sample. So collect the blood and then do a PCR from the blood sample. Then we have like a conventional PCR or real-time PCR. So what is commonly used in the diagnostic lab is a real-time PCR. And there are certain genes that will tell whether it is pathogenic or not. One example is LIP-L32, so which is commonly used. So that will tell us, that reading will tell us whether it is infection or not. So what is important here is the best option to catch them earlier for diagnostics is the using PCR. But it is not commonly practiced, especially in resource limited countries, because it is expensive. So if we want to do MAT, it only costs like less than 50 ringgit for one sample for few serovas. But if you want to do a PCR, it will cost like 350, 400 ringgit for one sample. So usually the PCR is done if we want to rule out certain infections. If we want to rule out lepto, then it is not like very primarily we ask them to do PCR because it is expensive. But in cases where it is really necessary, then we ask them to do PCR. So this molecular test can be done on blood, CSF, urine. Urine actually, again, it's not very good, not very good for the early diagnosis, because the lepto will be in the urine only in a later stage, because first it goes to the blood, right? From the blood only it will, like, after the kidney, and then later only it will go to the urine. So you can, because we do not know when the patient come to the hospital. He may be exposed to the infection many days back. Maybe his immune system is good and he's slowly trying to see the symptoms. But we do not know whether it is still in the blood or it has gone to the urine. So the best thing will be to get the specimen, like the blood is the best specimen for testing. So um, this we can do the conventional PCR. This is nested PCR, all in the research level. LAMP is another technique where you can detect the gene of interest, which is gaining very good attracting in it is being very much attracted in the diagnostics, because you don't need a PCR and it can be performed in resource limited settings. So LAMP is being tested for many diseases, infections, and we also have expertise doing LAMP in our department. So the genes that we can target is, there are a list of genes, but if you want to look for pathogenic, most of the time we use L32 gene. So our idea here is to check whether it is a pathogenic strain or a non-pathogenic strain. So pathogenic strain, so it should be a L32 positive. Okay. So laboratory diagnosis, from our experience of study, we found that if we do a early spiremic phase, that means 2 to 7 days of infection, then we do a PCR, and serology will work only like 7 to 14 days. So what we recommend is that whenever you suspect for lepto, you can do both the antigen detection and serology. Then you will be able to catch the patient, because we do not know at which day they are coming to the hospital, maybe on day 5 or day 7. So if they come like day 7, you will not be able to detect them by this, but maybe by serology. If they come very early, you will not detect by antibodies. Okay. So a little bit, so it is almost 11:52. So I will just go through a little bit on the public health challenges. Okay, this is picture taken from our own study. Maybe this is not, the first picture is not from ours, but most of the other pictures are from our own study. So we went to the different area, like the recreational areas, and some like crowded environment, or like, this kind of area, to see, like, we collected the environmental sample, soil and water, to see whether they have any leptospira inside that. So see, we can, this is not our picture, but I saw this picture. It's a very good example. See, they come to drink water from this recreational area, and then in return, they may urinate there, and that urine will contaminate the environment. Okay. So our data, this was studied and a long back, but I think it's more or less a similar now. This is not our study, but we did in 2016 to 17, 18, those areas. So they found that recreational area are the major, and residential area. So why this residential area? Residential area, because most commonly present in the crowded environment, where the houses are very crowded and they don't have the proper disposal of the waste, solid waste disposal, and then where the rats come. So it is like you're giving an area for the rats to come. So if we maintain this correctly, then we can reduce the cases. So this is from our study, that what we can see, that when we did, we tried to capture the rodents, the mammals, and this was done by our colleague in UKM, Dr. Farah. So we tried to see who are the people having lepto. So these are the different distribution. This ratus is a common rat that we see. The Novogicus and the ratus are the one. So the Novogicus is the one that we can see in the housing area. And at least 15% of these small mammals carry pathogenic Leptospira. And the species included interrogans HP358, Bok-Petersoni, Valley. So these three are highly pathogenic strains, and these three we isolated during our research on that. So we can see rodents everywhere, but at least 15% of them carry the Leptospira. So maybe to summarize, what are the current gaps and what are the future directions that we should focus? Leptospira infections are species and serovar specific. Hence, routine surveillance of human, environment, and animal is important to understand the dynamics of transmission and the appropriate intervention. So we need to do the surveillance, whether it is a recreation area or the housing area or the patients. We need to do this routine surveillance so that we have to know like how the Leptospira is transmitted from how they are transported or transmitted from one person to another person. It is usually not from person to person, but it is from area, like if like they are living in an area where more rodents, then we need to find what is the best intervention. Clinical diagnostics are not clear-cut as it mimics symptoms with several other illness, which delays the treatment and leads to severe leptospirosis. So, as I said earlier, we don't have a very specific symptom for lepto unless it is like in very severe leptospirosis. So several biomarkers are being invested for predicting the severe diseases. Hence, profiling the biomarkers and global evaluation is recommended. So recent studies show that there are so many biomarkers that will identify, that can predict Leptospira, but this varies also from country to country. And it is important that if we can profile these biomarkers and then do a global evaluation, then we will know like what are the biomarkers to look. Why it varies from country to country? Because the strains or the species in different countries are different in different countries. So it is important like what biomarker will be suitable for which species. So if we have that profile, it is a very, very good study that if we can profile that globally, then it is good. Uncertainty surrounds the effectiveness of antibiotic therapy during the immune phase of the disease when complication becomes evident. Systematic approaches, a clinical description of the disease, and community-based studies on leptospirosis transmission. So this antibiotic therapy, we are not sure like whether there is illness or not. So we're not sure whether to give antibiotic or not. But the good thing about Leptospira is like we don't see any resistance so far. So any like antibiotics that is given usually kills that is why most of the time we don't get the symptoms. And community-based studies are important for the transmission control. Clinical research on leptospirosis with a focus on patient care is scarce and needs to be prioritized in the tropical disease research agenda. So we need to know the clinical, so these are like ideas for future research, clinical research on leptospirosis. Okay. Then laboratory diagnostics are challenging due to the biphasic illness, the lack of diagnostic tool in resource poor settings where the disease incidence is high, leads to a diagnostic bias, favoring the identification of cases with classic symptoms. So new early point of diagnostics, point of care, and cost-effective diagnostics are crucial. So these are very challenging, especially like those who do not have the PCR facility and those who do not have like even resources to do the lateral flow or anything, or even the economically not supportive. So we need to find some early point of care and cost-effective diagnostics. Then people returning from recreational activities and sporadic cases from housing area are very common. So environmental transmission dynamics are important to be elucidated for appropriate intervention. So this part is very important because it is mostly we need to understand how it is transmitted in the environment. So we need to understand the dynamics. So studies have to focus on, because there are many sporadic cases from the environment, from the housing area, but we do not know how they are getting in. So we need to find that. And with my last slide, I just want to say the Malaysia will host the International Leptospirosis Society Conference in 2026, which is scheduled on 27 to 29 October 2026 in Putra Jaya Marriott Hotel. So this is for the first time will be in Malaysia, and it will be next year October. So please mark this in your calendar, and we will come with more updates and on the program and others. So I will keep you posted on the, because we just finalized the venue and the dates. So we are yet to finalize the speakers and things. So we will keep you updated. And when our website is ready, I will keep you updated. So please mark this. This is going to be a very interesting meeting, because International Leptospirosis Society, they do meeting once in 2 years, and most of the last year it was in Europe. So most of the times it is very rarely comes to like Asian region. It is very expensive to travel to Europe to do this, to see this or hear the, and it's a closed meeting. Like you will see all the experts in the leptospirosis research area will be in this meeting. Okay, so I think I will stop here. Thank you so much. Exactly at 12 o'clock. So thank you so much. I think we can take the questions. They said I can have more time. Okay, thank you so much for the opportunity.
Okay, thank you very much, Prof. And we have a few questions here from the participants. So hold on, I read it for you. So the first one is from one Susanna. So she would like to know, we have to subculture every 6 months to maintain. So how about the quality of the cultures? Do they maintain their potency and special characteristic, if any?
Yeah, any organism that if we keep on culturing, it will definitely lose its potency. But this cultures are, but we don't have any other option. But what we can do is like if we want to retain their antigenicity or potency or virulence, we can just take that culture and put into an animal, and after that we can isolate, I mean, get from that animal. So if we want to maintain the virulence, like if you want to get that extra, because since we cannot keep this in the -80 for many long years, we have to do the subculture every few months, every 6 months. So maybe after 10 subculture, if we see that it is not having the same characteristics as initial, it's very simple that we take this isolate and inject into a guinea pig, hamster, and then just collect the blood from that and isolate the organism from that. Then you will get back all these activities. Does that answer your question?
I hope so.
Okay.
So, the second question
Yes.
from Adia. So, she asked, is it necessary to have seroval genotyping done to determine the relationship between patient samples and environmental samples?
Typing to see the patient's and
to have seroval genotyping done to determine the relationship between these two samples, patient and environment.
So far, the genotyping of the serovar is not very important in terms of clinical diagnostics, because when it is infected, then you can treat them. But we also did some studies on different serovars on the hamsters, and we found that only some serovars are really like causing the pathogenicity, like interrogans. Yeah, yeah. So some syrup. So I think that has an impact on the for treatment, but maybe for research, yes.
Okay. So next question is a general question, Prof. So what is the lifespan of a leptospirosis bacteria in environment that can causes infection? I mean, how long will live outside the host to ready to get infected to human?
The pathogenic ones that comes from the animal that is excreted into the environment through the urine, that they will not stay for a long time, maybe a week to a month. And also, like when you can see that it is usually present in that if there is a wet environment, or like there is a little bit of water, they can live. But when the due to the sun, the UV can actually kill those lepto in the environment. Those environmental lepto, they can stay for longer time. But these pathogenic ones, which are excreted from the animal, they don't stay for longer. Like maybe like a week or a month or something like that. There was a study on. I did see that. I heard this in the conference, but I don't remember exactly, but it is not very long.
Okay, is there any recent findings that can relate to animal to human infections of leptospira? I mean, directly, not from their waste.
Animal to human, you mean the exposure directly from
Maybe saliva or something, or scratch,
yeah,
bites or
because
anything.
yeah, you can get the from the animal to the human, because if you are like very closely connected to the human, like if uh, like you're always with the animal, then you can get from the animal, because if you have a cut or burn or anything, you don't necessarily go to the environment. If they sit, you are like together with that animal for life, yeah, you can get that. But it is not like a transmission like a staph aureus or other things.
Mhm. Okay, understand. So the
Mhm.
last one is so what are the precautions and or red alert for the community in order to prevent the infection?
Keep the environment clean. The
Keep
best
the
thing is
environment
keep
clean.
the environment clean, so that you don't get rodents. And then if you go for any recreational activities or any water activities, after you come back, if you experience some fever or anything, tell the doctor that you went for these activities, so that they will be on the alert. And like those working with occupational risk, wear proper boots and others, so that you don't get the direct exposure to the environment.
Okay, thank you very much, Professor. If there's no more questions from the participants, I would like to close the Q&A session. So I hope this talk will be very fruitful for all. So our webinar session has come to an end. So again, I'm Jen, behalf of the Secretariat of PPSM Webinar Committees. We are grateful for your time spent in this webinar. So until we meet again in the next webinar.
Thank you
Thank you
so
very
much.
much.
Thank you.
Thank you,
Thank
Prof.
you so much.
Thank you all.